Glycobiology. Glyco means glucose. Biology means when glucose, generally glucose is present in the body of living organisms. Therefore, a study of glucose or a study of carbohydrate is called glycobiology. Okay? And as we know, when we say carbohydrate, I explained yesterday, hydrates of carbon or hydrated carbon. But carbohydrates are of three types: monosaccharide, sometimes oligosaccharide or disaccharide, and polysaccharide. When we say saccharide, saccharide means actually saccharide term derived from saccharum. You know saccharum? Saccharum officinarum? You don't know? It is botanical name of sugarcane. Saccharum officinarum. And as you know, the juice of sugarcane is sweet. Saccharum is a Latin word. Its meaning is sweet. Understood? So generally, carbohydrates, particularly monosaccharide and disaccharides, are sweet in taste, but polysaccharide is not sweet in taste. Okay? So we will study carbohydrate. Now, what is carbohydrate? Hydrates of carbon or hydrated carbon, because the ratio of hydrogen and oxygen is similar to water, that is 2:1. Present in plants as a starch and cellulose. Actually, a starch is the stored food material of plants. You might have heard. In our bodies, carbohydrate is stored in the form of glycogen, and the glycogen is present in the liver and muscles. What happens when we take food, and generally our food is rich in carbohydrate, and carbohydrate is digested into particularly rich in starch. Whatever we take, mainly we take plant products, and plants store carbohydrate, particularly starch. A starch is a polysaccharide, and by the action of different enzymes, particularly amylase, it digests amylose and amylopectin. Then it is broken down into disaccharide, and later on there is action of maltase. Then it is broken down into monosaccharide. So when it is completely broken down into monomers, then it is absorbable. When it is in the form of polysaccharide or disaccharide, it cannot be absorbed. Therefore, there is need of digestion of polysaccharides and disaccharides. Sometimes there is a question, actually it is reasoning type, which carbohydrate is not digested, or which carbohydrate is digested. If there is a question and there is option, fructose, maltose. Suppose one question is not digested: fructose, maltose, amylose, or what will be the answer? Fructose, because fructose is monosaccharide. So sometimes because monosaccharides are simple, small and diffusible, therefore there is no need of digestion of monosaccharide. Understand? So a starch is converted, particularly in our bodies, or enzymes present or enzymes secreted by different salivary glands are able to digest the starch, but cellulose is not digested by animals, particularly by human. It is digested by animals. You might have heard wood roaches and termites, because in their salivary gland there are certain symbiotic bacteria that secrete cellulose, and that's why they are able to eat even wood. Wood contains mainly cellulose. Okay. Particularly when we take cellulose, when we take vegetables, green leafy vegetables, then it acts as laxative, constipation remover due to presence of cellulose. Because cellulose, as you know, particularly in rainy season, doors of the room does not close properly. Why so? Moisture is absorbed. The cellulose has the property to absorb the moisture. It means absorb the water. So what happens when there is lack of water only, then there is constipation. When that cellulose absorbs water, then it acts as laxative. Understand? So nowadays, as you know, cellulose is being treated, and after treatment, there is fibrous food. It contains different types of. As that day we went to VSI, and they are making dietary fibers from the molasses of sugarcane. It means they are making, or you can say, they do processing of the cellulose, and then it will be used as laxative or dietary fibers. It will help in the normal activity of the digestive system. And animals has glucose and glycogen. In our blood circulation, generally 80 to 100 milligram per 100 milliliter of blood glucose is present. Level of glucose in our blood is 80 to 100 milligram in the normal condition. You might have observed sometimes we feel weakness, and sometimes we feel we will fall down. At that time there is lack of glucose. Particularly those blood vessels that supply blood to brain, heart and lung, because these are fundamental organs of our, or you can say vital organs of our bodies. There is need of pure oxygen and sufficient amount of glucose. When there is lack of glucose, or you can say when there is hypoglycemia, glucose level becomes low, then we feel weakness, and sometimes we feel we are unable to even stand. So that is glucose. Therefore glucose is called blood sugar. And when we take food, after taking food, it goes up to 180 milligram per 100 milliliter of blood. When it goes 180 milligram per 100 milliliter of blood, then where it goes? So after absorption, it goes in the circulation, but extra amount of glucose is stored in the liver and muscles. What happens when blood glucose level is high? At that time, insulin is secreted by the beta cells of pancreas, and it affects the polymerization, or you can say it increases the rate of polymerization or glycogenesis. Polymerization of glucose in liver and muscles is called glycogenesis. There is formation of glycogen, and it is stored in the liver, and some amount is stored in the muscles also. Okay, but our brain or heart does not store, isn't it? Muscles store, liver stores, but brain or heart does not store. Why not? Actually, these are vital organs. Wherever there is a storage, when there is formation of glycogen bodies, you might have seen along with age there is shrinkage in skin and muscles, becomes loose. wherever there is a storage, it affects the metabolic activity or function of the cell. So if it stores in the nerve cell or stores in the cardiac muscle, then its activity will be affected. If these cells are unable to do normal functions, then what will happen? But in muscles, if for some time, if we feel tiredness, we feel tiredness in the muscles of body, hand and feet, but never we feel tiredness in cardiac muscle. Do we feel? Why not? Different types of muscles are present in it. Triceps, biceps, latissimus dorsi, femori. Yeah, all these muscles are present in our bodies, and particularly body muscles or limb muscles feel tiredness, but cardiac muscle does not feel. Why so?
Lactic acid accumulates in the muscles and we feel.
Why lactic acid accumulates in muscles?
Anaerobic respiration.
Why anaerobic respiration?
Shortage of oxygen
Proper oxygen supply is not present.
during running or like.
When we do extra work, whatever oxygen goes to the muscles, if we do more work than that, and as you know, our muscles contain myoglobin. Myoglobin stores oxygen. Hemoglobin transports oxygen, and myoglobin stores oxygen, and like that, you might have heard lactoglobin. Have you heard lactoglobin? No. Lactoglobin is present in the leguminous plants, in the root nodules of leguminous plants. You might have heard, particularly pulses. Yeah, root nodules of pulses, different type of pulses, they are able to synthesize or they are able to fix the atmospheric nitrogen. And at that time there is need of anaerobic condition. Those bacteria cannot tolerate aerobic condition. So whatever oxygen is present in the root nodules in leguminous plant, that oxygen is trapped by the lactoglobin. Therefore, in the root nodules of leguminous plant, lactoglobin acts as oxygen scavenger. It cleans. Okay? If oxygen will be present there, then bacteria will die. They will not able to survive because they are obligate anaerobes. Understand? So that's why. So what happens when there is extra amount of glucose? It will be converted into glycogen. And one more thing is there. When we take food, we feel strength, isn't it? As I mentioned yesterday, is some psychological effect, not only psychological effect. Of course, psychological effect is there. We think, and then our endocrine glands start to act. One of the part of that endocrine gland is secretion of insulin also. When blood glucose level becomes high, and when it reaches to, as you know, for the entry of the glucose inside the cell of, when it circulates and its level becomes high, so at the time of, suppose this is your beta cells of islets of Langerhans of pancreas. Not only in beta cells, in almost all cells of the body, there is glucose transporter. And this glucose transporter depends on, actually transport through the glucose transporter dependent on the concentration of insulin. Insulin affects its, so what happens, suppose this is glucose transporter. When insulin level becomes high, because due to intake of food or due to digestion of food, carbohydrate is digested, glucose level will go up. So due to that there is secretion of insulin, what happens when the glucose level becomes high, suppose it reaches in the tissue, at that time through glucose transporter, more amount of glucose enters inside the beta cells. Then beta cells feel that there is extra burden of glucose in the blood and it should be controlled. Then, as you know, there is signaling, and ultimately there are two types of effect. One thing is whatever vesicles that contains insulin, they are released, and another at molecular level. The gene responsible for insulin is also upregulated or activated. Understand? Then this insulin will bind with the different cells, and wherever there is deficiency or lack of, because this glucose transporter is, you can say, recycled. It goes inside the cell and it comes, actually when it is degraded and again new glucose transporters are constructed. So it will affect other cells and new types of glucose transporters are recruited in the cell, and there is more entry of glucose inside the cell, and then there will be more amount of glucose inside the cell, so they will be oxidized and more energy generation. And when more amount of glucose enters inside the hepatocytes, because glucose, number of glucose transporter in hepatocytes and muscle cells is more in comparison to other cells, because other cells require only a particular amount of glucose for energy generation to complete its physiological activities. But hepatocytes and muscle cells store glycogen. Therefore, there should be more amount of glycogen, more amount of glucose should enter inside it. Therefore, there is more number of glucose transporter in hepatocytes. Understand? So all these things are correlated. So thus, glucose enters inside the cell. In normal cells, it is oxidized and energy is generated. But in liver and muscle cells, besides energy generation, they are stored in the form of glycogen. Okay? Now, it is most abundant biomolecules on the earth, as you know. Plants, cellulose, the most abundant part, isn't it? So it is present in the human bodies, it is present in plants, it is present in insects in the form of chitin. So it plays mainly two roles. One role is energy generation. One function of carbohydrate is energy generation. Wherever processes or whatever physiological activities, initially glucose is utilized for the generation of energy, and another is formation of certain structures. Of course, in human body, it takes part in the formation of certain receptors. You people have studied. Many receptors are glycoproteinous in nature. It means it is attached with the protein, and particularly when we determine blood group, that antigen is also glycoproteinous in nature. But in insects, that you ask me that why cockroach is surviving till now, there is no single. The main thing is chitin. Chitin is also a carbohydrate. And in plants, cellulose. Understand? So these are important functions of carbohydrates. Now, classification of carbohydrates.
Sir, is there any particular place that plants will store that cellulose and starch?
Yeah. Actually, when you say plastid, you know plastid? Plastids are of three types. Have you heard? Leucoplast, chloroplast, and chromoplast. What is the function of leucoplast? Storage of starch. And there are many types of ileoplast, many storage of lipid. Why? Okay, so that is function. When it is chloroplast, then chlorophyll is present, it will do the function of photosynthesis. Particularly those tubers. Tubers are rich in leucoplast. They contain a large amount of starch, carbohydrate. So on the basis of units, carbohydrates are divided into 3 groups. Actually I have put monosaccharide, disaccharide and polysaccharide. But if you go chemistry point of view, then you will mention monosaccharide, oligosaccharide and polysaccharide. Why I have put it? Because biologically disaccharide or tetrasaccharide or pentasaccharide or hexasaccharide or heptasaccharide are not important. Understand? When we say oligo, oligo means few, 2 to 20. If 2 to 20 monosaccharides are together, they are called oligosaccharide. Disaccharide is also a part of oligosaccharide. But biologically, all oligosaccharides are not important. Understand? Therefore, I haven't put oligosaccharide. For your concept, you have to remember 2 to 20 monosaccharides together with are called oligosaccharides. Okay. So when there is only one unit monosaccharide, 2 units disaccharide, and more than 20 units polysaccharides. Okay. I thought today I will describe whole carbohydrate, but it is very difficult because I was studying from different books, and each and every book is telling something else. And in these books I found many mistakes, particularly in these books. There are many errors. When I was reading it, then I found particularly these are not matured persons. They do not go in depth. They copy sometimes from here and sometimes from there. They should write book in a proper way. They mentioned D form, and later on they are writing laboratory. What is this? D form is dextrorotatory. They are mentioning D form minus sign, and they are and the next page about the same carbohydrate they are mentioning laboratory or labulose. We say dextrose or labulose on the basis of its we will study on that. So monosaccharides, when there is only one unit, therefore they are called simple sugars because they cannot be hydrolyzed into a simple form. As I mentioned, during digestion, what happens? Hydrolysis. When there is sucrose, in the presence of sucrase enzyme, sucrose is broken down into glucose and fructose. When there is maltose, in the presence of maltase, it is broken down into 2 molecules of glucose. When we say lactose, lactose is broken down into glucose and galactose in the presence of lactase enzyme. So all these enzymes add 1 molecule of water in the disaccharide, and then disaccharide is broken down into monosaccharide. Okay. So that is hydrolysis. Empirical formula CH2O whole N. Simplest monosaccharides are trioses. Actually, particularly monosaccharides are named on the basis of number of carbon present in that monosaccharide. When there are 3 carbons, they are called trioses. When there are 4 carbons, tetroses. When 5 carbon, pentoses. When 6 carbon, hexoses. When 7 carbon, heptoses. In many books, people have mentioned octoses, nanoses, decoses, but we will study only up to maximum up to heptose, not more than that. Heptose is also not important for our purpose, but our purpose is up to hexose, different forms of glucose, galactose, fructose, sucrose, psychose. We have to study only those things. So now forms of monosaccharides on the basis of alignment of the H and OH groups around the penultimate carbon atom. Actually, when we write, this is the functional group. This is the functional group, and if we break it to this form, CH, C, double bond. This is our glyceraldehyde, isn't it? This is our glyceraldehyde. This is CH2OH, CHOH and CHOH. This is glycerol. Initially it was glycerol. When it was removed by, when it was replaced by aldehyde, then it is glyceraldehyde. Now we start numbering from the functional group. It is first carbon, it is second carbon, and it is third carbon. Okay, like this, if we
So functional we can write from the downwards also. Means we can write in 1, 2, 3 from downwards also.
how? This is for hexose. It will be what you were telling.
So we can write, means we can count the UPEC name from the down also. We are telling that. From the bottom.
From where did you start numbering? From the functional group or from anywhere?
From the functional.
Then how you will start from bottom? Here, this is the aldehyde, okay? And here is aldehyde, okay? So we will start first carbon, second carbon, third carbon, fourth carbon, fifth carbon, sixth carbon. Last carbon is sixth carbon. Before the last is called penultimate. Before the last is called penultimate. If at the penultimate position hydrogen is present in the left side, then it is dextrorotatory. When plane polarized light is passed through the solution of a particular type of monosaccharide, where at fifth carbon hydrogen is attached or hydrogen is present in the left side, then it is called deform or dextrorotatory or dextroge. Understand? If it is just opposite, it is and like this, then it becomes levorotatory. And particularly in nature, you people might have seen, always we take D-glucose. Almost all natural forms are present in the D form, not in L form. But there are certain exceptions, L-mannose and L-rhamnose and L-dopa. L-dopa is used for the treatment of Parkinson's disease. That is in the L form, not in the D form. When D form is given, cells are not repaired. So most of the natural form is this is our D form. Okay. The D form depends on the position of hydrogen on the penultimate carbon. What is the position of hydrogen in the penultimate? If it is in the left, then it is dextrorotatory. If it is in the right, then levorotatory. When it is in the left, then it is written minus D or minus. And when it is in the right, then it is L plus. The symbol will be plus.
So isn't it based on OH group?
Actually, in many books they have written H and OH both. But I checked in 2-3, you can say, good books. Rather, at night I was reading a book. Perhaps it was Steyer and one more. So in the Steyer they have written both H and OH. I have to check in Lehinger, because Lehinger I did not check what is the exact Lehinger I mentioned. But there was another book in which they have mentioned only position of H, not OH. They have
You
mentioned.
remember studying it on the basis of OH.
OH.
OH on the right is D. It's the same thing. OH on the right is
If it is there, if you mention H or AOH, therefore many people have mentioned both H and OH. If H is in the left,
OH.
OH will be in the right. So you can consider both. But as I found in a particular book, only H. If they might have mentioned only OH, then I mentioned that. But they have mentioned only H, therefore I have. But if you change the position of H, the position of OH will change automatically. Therefore you can consider both. Okay. So, therefore alignment of the H and OH groups. I have written, but in that book they have mentioned only H. Groups around the penultimate carbon atom. Now, forms of monosaccharide on the basis of alignment of the hydrogen around the penultimate carbon atom. D form, when H is on the left and OH on the right, and just opposite is L form. So that is dextrorotatory and it is levorotatory. Whatever form is D form, they are called dextrose. When it is D form, dextrose. When it is L form, levorose. Now, on the basis of projection, actually there are two types of projection you might have studied: Fischer projection and Haworth projection. Fischer projection was given by Emil Fischer for open chain, and Haworth projection was given by Haworth for cyclic structure. And particularly, what is the difference? Fischer projection, there is two-dimensional representation of three-dimensional molecules. Actually, when you put a molecule, suppose you want to show this H and OH towards the reader or towards ourselves, and this H and this bond towards the board. Understand? So if it is represented by a specific lining, you people might have seen, if it is solid line and dashed. Yeah? So if solid line and dashed line, that is Fischer projection. And it is in the form of, it is three-dimensional molecule, but it is represented in two-dimensional molecule, because on the paper it is not possible. Therefore, for open chain, Fischer projection was proposed. And for ring structure or cyclic structure, Haworth projection was with a simple three-dimensional perspective. Now, on the basis of number of carbon in the ring, actually, particularly that ring form is particularly in hexogens. When we say hexogens means 6 carbon, pentogens means 5 carbon. In hexogens, the ring is present in two forms. furan ring and pyran ring. You might have heard, no? Furan ring and pyran ring. When in the ring there are 5 members, 1, 2, 3, 4 and 5. When there are 5 members in the ring, 4 are carbon atoms and 1 is oxygen. Then it is called furan ring. And when this is pentagonal ring, this is pentagonal ring and it is hexagonal ring. Hexa means 6. When 6 membered ring, but 5 are carbon and one atom is oxygen. This is pyran ring. So as we know, glucose and fructose. Glucose is hexose and fructose is also hexose. But glucose is pyranose and fructose is furanose. Understand? Because in the ring form, their structure is different. Only 4 carbons are present in the ring in the case of fructose. In the case of glucose, 5 carbons are present in the ring, while the molecular formula is the same. Understand? So therefore, furanose, wherever there is pentagonal structure and 4 carbon atoms are present in the ring and 1 oxygen atom is there, such a monosaccharide or such a hexose is called furanose. And when hexagonal structure, 5 carbon atoms and 1 oxygen atom, then it is called, such a hexose is called pyranose. So glucose is a pyranose and fructose is a furanose. Now on the basis of functional group. If functional group is aldehyde, then it is called aldose. If functional group is ketone, then it is called ketones. Here functional group is aldehyde, then it is called aldose. Glyceraldehyde, in glyceraldehyde functional group is aldehyde, therefore it is aldose. But in dihydroxyacetone, functional group is ketone. You can break it in this form. Here, this is functional group. Their functional group is ketone. Their functional group is aldehyde. So this glyceraldehyde is aldose. Functional group is aldehyde. Ose is mainly for carbohydrate, or you can say for monosaccharide and disaccharides. We use OH because for chitin or for glycogen we do not use OH. And whenever monosaccharide and disaccharide, either glucose, fructose, mannose, ramenose, glucose, sucrose, then maltose, sucrose, lactose, all these are monosaccharide and disaccharides. So for monosaccharides and disaccharides we use OH. So wherever, actually we are studying about monosaccharide. If in a monosaccharide the functional group is aldehyde, therefore we call it aldose. Aldehyde plus OH, aldose. And if functional group is ketone, then ketose. So our glyceraldehyde is aldose and dihydroxyacetone is ketone. Like that, our glucose is aldose and fructose is ketone. Okay? So now we will study all these things. Now, types of monosaccharide on the basis of carbon atoms. Number of carbon atoms. Now, how many carbon atoms are present in a particular monosaccharide? On that basis, we have triosage. It means number of carbons will be, here you can count it, 1, 2, 3. Dihydroxyacetone. In glyceraldehyde also 1, 2, 3. So dihydroxyacetone and glyceraldehyde both are triosage, but one is aldose, another is ketose. Now you can see diglyceraldehyde. You can see at penultimate position, at penultimate carbon, H is present in left side, then and OH in right side, then it is D, and just opposite of it is L-glyceraldehyde. Understand? But in the nature it is abundant. L-glyceraldehyde is not found. Where trioses are present, particularly they are formed in the body during glycolysis. When we say glyco, as I mentioned, glyco means carbohydrate or glucose. Whatever glucose, they are carbohydrate. So glyco means glucose and lysis means breakdown. Breakdown of glucose is called glycolysis, and it occurs in the cytoplasm of the cell. It occurs in the cytoplasm of the cell. One more thing. Actually, for anaerobic and aerobic respiration, for both respiration, glycolysis takes place, isn't it? Now, tetrosage, 4 carbon containing sugar, therefore they are called tetroses. Its example is erythrose. Is an intermediate of pentose phosphate pathway. Particularly pentose are ribose and deoxyribose. They are ribose, ribulose, deoxyribose. They are pentoses. It means wherever there is synthesis of nucleic acid. In the nucleic acid, in DNA there is deoxyribose. In RNA there is ribose. So wherever there is synthesis of DNA or RNA, there is need of pentose phosphate pathway, and as an intermediate of that pentose phosphate pathway, erythrose is present. It is an intermediate of that pathway, erythrose. Then triose is a degradation product of ascorbic acid. Ascorbic acid is vitamin C. When it is degraded, then there is generation of triose. Triose is also a tetrose. Now, all erythrose and triose both are, you can see, erythrose and triose. Functional group is what? Aldehyde. De-erythrose, de-triose. Functional group is aldehyde. Therefore, both are aldoses. While erythrulose, functional group is ketone in both D and L form. Functional group is ketone. Therefore, erythrulose is a ketose. Present in raspberry, that is raspberry. Therefore, I have it. Now, pentoses. They are important constituents of nucleic acid, as I mentioned. DNA contains deoxyribose, RNA contains ribose. And many coenzymes, you people might have heard NAD, NADP, and energy currency, ATP. ATP contains what? ATP is a nucleotide where 3 phosphates are present. It contains nitrogenous base, sugar and phosphate. Phosphate are 3 in number. So therefore, coenzymes NAD, nicotinamide adenine dinucleotide, NADP, nicotinamide adenine dinucleotide, phosphate. Therefore, it's coenzymes. Ribose present in RNA and deoxyribose in DNA. Then you can see it is deoxyribose. Functional group is aldehyde, and this is ribose. In ribose also functional group is. What is difference between DNA and RNA? Why it is called DNA? What is the basis?
Sugar, one
Sugar.
oxygen.
When it is deoxyribose sugar, then it is called DNA. In RNA there is ribose sugar, therefore it is called ribonucleic acid. Where is the difference? First, second. You can see deoxyribose, it is CH2. This oxygen is not here. In the case of deoxyribose, at second position, there is no oxygen with H. Understand? There is only hydrogen here. In the case of ribose, therefore formula of ribose is C5H10O5, and formula of deoxyribose is C5H10O4.
So, where is that enzyme FAD used? It is also coenzyme, right?
Yeah, FAD, flavin adenine dinucleotide. Particularly when succinate dehydrogenase in the Krebs cycle. At that time, that electron is proton is accepted by FAD. No? Then and whatever FAD from FAD H2, there is generation of how many ATP? 1.5. And from NADPH2, 2.5.
So 3 MPD.
That passes. Now you will search. In science, nothing is constant. You know, I became surprised. Once I was sitting in an interview and I was asking question. Okay, from one NADPH2, how many ATP is generated? The student right answer, 2.5. 2 and 3 are old concepts. 1.5 and 2.5 are new concepts. And when a student, actually he was emphasizing and I was just scolding him. You don't know these things? Then he told, sir, it is new concept. And when I checked it, it was very embarrassing for me. When I checked it in white and white, and then I checked on different, initially I checked on net, then I checked in white and white, recent edition, I found this is the. So therefore in science, at that time I felt, how much science changes. Each and every person. And particularly, therefore it is told that a teacher of science should always be a reader, not a teacher.
So then ATP generation number will also change.
Of course.
It will change.
Yeah. They have mentioned clearly, they have given a region, how many protons are taken from the mitochondrial matrix to the outer chamber, and when they return, how many return, then one ATP is synthesized. That is the basis, and they have actually what happens in science. Therefore we say, whatever we see, that is not the world, isn't it? Bacteria, viruses, different pathogens, they are not visible to us. But they exist or not? When we observe with microscope. And as much advancement in technology, along with that, our observation capacity increases. So along with that, therefore I think where is it? Isn't it? Initially people were observing on simple microscope, then compound microscope, then phase contrast microscope, then electron microscope, then SEM, scanning electron microscope, then TEM, tunneling electron microscope, now STEM, scanning tunneling electron microscope, now AFM, atomic force microscope. What is that? Is it? Now it is over? No. We don't know. Next step will be what? So when they were measuring, so initially they were on that, whatever instruments they measured, they calculated that membrane potential. But whatever instruments now developed, it is more sophisticated than that. And it tells the exact membrane potential. How many protons are transported, then how much membrane potential is generated, and that's why there is again movement through proton channel in the oxysome. On that basis they have calculated, and they have given its explanation, and that is more logical than prior one. Therefore, it is most accepted. Now it is most accepted. Now, arabinose, what we were discussing. Okay. Now I would like to. What is it? Huh? Of course it is pentose. Of course it is pentose, but what pentose? Ribose. It is. And when we remove, now it is deoxyribose. From here we start numbering. First, second, third, fourth, and fifth. At second position, therefore we write. Prasad, do you have any objection? 2' deoxy. At second position, D means decrease, oxy means oxygen. At second position, there is loss of one oxygen, then such a pentose is called deoxy. When oxygen is present there, then it is ribose. Both are pentose because number of carbon is 5, only there is change in number of oxygen. On that basis, it is deoxyribose or ribose. Okay? Now, arabinose and xylose are present in plants. You can see arabinose. It is also what? Aldose. Xylose, aldose, lexose, aldose. Particularly, you people might have heard hemicellulose. Have you heard? Hemicellulose is made up of arabans and xylans. Have you heard? No. You haven't heard? Huh? Arabans and xylans? Arabans and xylans. Particularly, when there is many units of monosaccharide, then it is called glycan. In one word, you can say, for glucose, for cellulose, for starch, for glycogen, for chitin, for hemicellulose, you can say only one word, glycan. Understood? It means many monosaccharides together, or polymer of monosaccharide is called glycan. When many arabinose is a pentose. When many arabinose is together, then it is called araban. Okay? And when many xylose together, it means polymer of xylose, then it is called xylan. So araban and xylan are present in hemicellulose. Therefore, I have written it in plants. In hemicellulose, and as you know, hemicellulose is present in the cell wall. Lexose present in bacteria in a small amount, and ribulose is involved in photosynthesis. You people might have heard many times this question I have faced. RUBP, most abundant protein on the earth. Most abundant protein on the earth is rubisco, ribulose biphosphate carboxylase, isn't it? Ribulose biphosphate carboxylase is the enzyme, and as you know, enzymes are chemically protein, and it is most abundant in the nature. Because wherever photosynthesis takes place, rubisco is required, and there it acts on ribulose. So ribulose is involved in photosynthesis. Therefore, so you can see here it is ketone group. It means ribulose is ketose. Understand? You have to remember BOJ aldehyde, ulose ketose. Understand? Only OJ aldehyde, ulose ketose. Okay? Now, exogens. They are physiologically most important monosaccharides, isn't it? As you know, when there is photosynthesis, formation of or synthesis of glucose. When we take food, energy is generated by the oxidation of glucose. That's why it is most important physiologically. And now glucose. The first is glucose, also called blood sugar or corn sugar or grape sugar. Yeah? And normal blood glucose level is this instantaneous source of energy. As I mentioned, there is no need of digestion, and it is directly absorbed, and then it goes in the circulation, and then it goes to the tissue fluid. You know, it is transported by blood and reaches to the tissue, and it enters in the cell because glucose transporter is present there, and then it is oxidized and there is generation of energy. Stored in liver and muscles as glycogen. Sweetening power is 74. People might say, nowadays I take glucose, but it does not so much sweet as when I take syrup. When I take solution of sugar, that table sugar, it is not so sweet, but it gives energy. It is not so sweet, therefore sweetening power is 74. Particularly for sucrose, sweetening power has been considered 100. So it is, in comparison to sucrose, it is 74. Understand? Enters inside the cells due to insulin. As I mentioned, when insulin is secreted, then there is effect on the glucose transporter, and then it enters inside the cell. That's why when there is deficiency of insulin, there is diabetes mellitus or insipidus. Then when insipidus occurs,
receptor will be damaged.
Huh?
On the cell surface, the receptor will be damaged.
then it will be
So due to ADH hormone
deficiency of ADH, antidiuretic hormone. Yeah?
excessive
Then there's-
urination.
Huh?
Excessive urination.
Yeah, polyuria. Micturition becomes more frequent, and that is in the case of polyuria. And there is polydipsia, more thirst and more urination. Upernal niche. So that is the case of polyuria or polydipsia. That is due to deficiency of ADH. ADH acts on the distal convoluted tubule, and when there is deficiency of ADH, water is not absorbed, and whatever water is filtered from blood, it goes out. That is the case of polyuria. And in the case of diabetes mellitus, diabetes mellitus is of two types, NIDDM and IDDM. NIDDM is non-insulin dependent diabetes mellitus, when that receptor is damaged. Particularly in children, if our receptor is not present. When in children, the gene for glucose transporter is mutated. If insulin is sufficient amount, but transporter is not there, how glucose will enter? You give the injection of insulin, besides it does not enter inside the cell, because that disease is not dependent on insulin. That glucose transporter is damaged. Understand? So that is called NIDDM, non-insulin dependent diabetes mellitus. Okay. Another is IDDM, insulin dependent diabetes mellitus. Particularly when there is less concentration of insulin. You people might have heard, particularly in old people. In old people, there is less concentration of insulin, particularly beta cells, become degenerated, and people have reported that some proteins present in the milk, it damages beta cell. And alloxan is also a chemical. We will write it because alloxan and streptozotocin. When we develop the model of a diabetes, animal model, so what is done? Either alloxan or streptozotocin is injected in animals. When it is injected in animals, then it destroys the beta cells. And when beta cells are destroyed, then what will happen? Insulin will not be secreted, and when insulin is not secreted, glucose will not enter inside the cell, and then there is hyperglycemia, glucose level, and the individual becomes weak, because when glucose does not enter inside the cell, there is no generation of energy, and when energy is not generated, glucose does not enter, therefore it remains in the blood. Animal takes food, it digests, blood glucose level becomes high because it is not entering inside the cell. Understand? So that is due to that. Now, one molecule of glucose produces 686 kilocalorie energy. Actually, when I was studying, this data was also different. It was 673. When I was studying, it was 673, and later on people calculated and they mentioned 686 kilocalorie. I have taken it from a book. Therefore, you people will check it. Whenever I mention data, don't be blind, particularly in CSIR. Question setters are scientists, and scientists of that particular, they are expert in that field. Understand? So therefore, when you are going to appear at the exam, up to that time, you should be ready, particularly whatever new things are coming. If something is new, some Nobel Prize is given in biology field, so you have to keep those things in your mind. Because sometimes they ask very recent, whatever new research is there, at least 10 questions are very recent. If you do not read journals, or if you do not update yourself with the recent development, then you will not be able to touch those 10 questions. You can qualify because there is option, but if you get, then you can be an SPM. You can be a higher ranker, you can go for a Shyama Prasad Mukherjee fellowship. So therefore you have to see. So here it is all the, that is ring, that is open chain of glucose, and these are alpha D and beta D. Actually, what is the basis of alpha and beta? When this first carbon, if OH is towards downward, then it is alpha. If OH is towards upward, then it is beta. And normally, in the nature, beta form is abundant. Why it is abundant? Because when it is three-dimensional structure, then this OH goes towards this, and then there is repulsion between OH and OH. Therefore, it prefers more, it goes away from this. Therefore, in nature, mainly beta form or beta D glucopyrogenate is pyran ring. It is pyran ring. It is hexagonal ring. Therefore, it is pyranose and it is glucose. So, therefore, it is alpha D glucose and it is beta D glucose.
One second.
I explain it here. and sixth. There are 6 carbons. Therefore, it is hexose. Functional group is aldehyde. When you see in the open chain, it is aldehyde. That's why it is aldose. So it is aldose. It is hexagonal ring. Therefore, it is pyran ring. And in pyran ring, 5 carbons are in the ring, and 1 carbon is outside, and 1 atom in the ring is oxygen. So therefore, it is aldose pyranose. Okay, it is glucose. C6H12O6. It is glucose, and at the first position, if OH is towards downward, then it is alpha. Then it is alpha. If this OH is towards upward, then it is.
Sir, one second. You said when we consider the formula C6H12 and O6, but when you was bifurcating that furanose and pyranose, you consider only 5 carbon for glucose. You give the
5 carbon in the ring,
so
and 1 carbon is out.
classification is based on the ring.
Huh?
In this classification, then it's based on the ring. So you're
That
considering 5
classification,
carbon.
the 2 forms, 2 rings mainly, the 2 forms furanose and pyranose. That is pyran ring and furan ring. In pyran ring, 4 carbons and 1 oxygen. That is pentagonal ring. And 2 carbons are outside the ring. So how many carbons total?
6.
6.
So it's based on the ring. This classification
Yeah,
is based on the
based
ring.
on the ring. When it is pentagonal ring, that is furanose. When it is hexagonal ring, then it is pyranose. Understand? So this is alpha D glucopyranose. It is glucose and it is pyran ring. Therefore, it is alpha D glucopyranose, or you can say alpha D glucose. No need to write pyranose, but you have to know it. It is pyranose, and that in the case of fructose, it is furanose. So you can see alpha D fructofuranose. Understood? So that is on the basis of that ring only. Okay. That is your glucose. Should I stop it here?
Which is like abundant,
Huh?
which is more available in nature.
Beta form. As I mentioned, when that repulsion occurs. Should I stop it here? Because I have to do. One person is calling repeatedly. And next was, let's see how many slides I will take now. Manoj. Obtained on hydrolysis of plant manoscience and gums. These are present in plants, not in all plants, some specific plants, I forgot that name, in the specific plants where it is present. Mannitol. Actually, when this alcohol, when this sugar is converted into alcohol, understand? When we say O's means sugar, and when we say all means alcohol. When it is manoj, it means it is sugar, and when we say mannitol, understand? So mannitol is a sugar alcohol produced by reduction of mannose or fructose. Mannose and fructose both are hexose. Both are hexose. Therefore, from any it can be generated, able to loosen the hold of bacteria with kidney, ureter, and urinary bladder. Therefore, particularly for urinary tract infection or kidney infection, mannitol is given. Understand? Because wherever the bacteria, as you know, for the infection of the parasite with a particular type of cell, there is need of interaction, isn't it? The parasite binds with the host cell, and then there is host-parasite interaction. And only then there is infection. So when it is adhered, the bacteria is adhered with the cells of all these parts, kidney, urinary bladder, if mannitol is given, it removes, or you can say it detaches them. It detaches bacteria from the urinary or excretory system. Therefore, for urinary infection, mannitol is given. And another is crosses the blood-brain barrier. As you know, to treat the nervous system disease is very difficult because most of the drugs do not cross the blood-brain barrier. But mannitol crosses it. Actually, due to mannitol in the blood-brain barrier, there is constriction of the endothelial cells. In the blood-brain barrier also there is blood capillaries. There is blood capillaries, and in that blood capillaries, whatever endothelial cells are present, in the presence of mannitol, there is constriction, or you can say shrinkage in endothelial cells, and the tight junction also becomes loosened. That's why it is able to cross the blood-brain barrier. So therefore, it can be used as a vehicle for the transfer of, or you can say therapeutic drugs for the nervous system related disease, and used in osmotherapy, because it constricts and relaxes of the constriction things is involved. That's why it helps in the osmoregulation also. So this is your mannitol, because it is neither aldehyde nor ketone. Here is OH group. Okay, here it is aldehyde, therefore it is mannose, and here there is OH, means all alcohol, therefore it is mannitol. Now, galactose, also called brain sugar, because it is utilized by the neurons or nerve cells. Most rapidly transported monosaccharide, synthesized in mammary glands. As you know, lactose is the milk sugar, and lactose is made up of glucose plus galactose. So glucose is present in the blood, but galactose is not there. So it is in the milk. When there is need of synthesis of milk, at that time glucose and galactose both are present, only then there will be formation of lactose, a constituent of milk sugar and agar. If you know agar, you prepare that what? Gel. From where it is obtained? Huh? Seaweeds. What are those seaweeds? From which family? It is from algae. Isn't it? What is the family of that algae? Rhodophyceae. Rhodophyceae are red algae. And a specific genus. What are those genus? Gelidium and Gracilaria. From Gelidium and Gracilaria, agar is obtained. That is your, that belong to Rhodophyceae. You have to remember all these things. No? It is small things, but both, because it is involved in all experimental things. Gelidium and Gracilaria. G-E-L-I-D-I-U-M, Gelidium, and Gracilaria, G-R-A-C-I-L-A-R-I-A, Gracilaria. Now, fructose, also called fruit sugar. It is a furanose. You can see here. Second, third, fourth and fifth. Only 4 carbons are present in the ring, and fifth atom is oxygen. It means this ring is furan ring. 2 carbons, first carbon and 6th carbon are outside the ring. Total number of carbon is 6. They are hexoch, but 2 carbons are outside the ring. So the whole ring is pentagonal. It is pentagonal ring, and 4 are carbon atoms and 1 is oxygen atom. Therefore, it is furan ring. So therefore, it is a furanose, present in fruit juices except in grapes and corn. In grapes and corn it is not present. Honey contains highest amount of fructose. Therefore honey is sweetest. And sweetening power is 172. If you take sugar, cane sugar, and if you take fructose, the fructose is around 2 times, or you can say 1.7 times sweeter than your normal table sugar. Therefore, sweetening power is 173. Therefore, it is sweetest sugar. Whatever carbohydrate present in the nature, the sweetest carbohydrate is fructose. And you might have heard, particularly those people that suffer from diabetes, they take saccharin for sweetening, because it is around 300. Its sweetening power is around 300. And now it is, you might have heard monoline. Monoline is, I haven't heard. What is sweetening power?
So the sweetening power was mentioned in Dinesh.
Oh, when I was studying at that time, I found in 2002 or 3, monoline is a protein, and its sweetening power is more than that of saccharin, around 1000, while saccharin is around 300. Like that. Exact, it is not exact data. No? I am recalling whatever I am telling you. So particularly, people have, by research, people have suggested that those people that suffer from diabetes, they can take monoline as substitute for table sugar or cane sugar. Source of energy for sperm in semen. As you know, when there is spermatogenesis, when complete sperm forms, and at that time, when there is, as you know, in the semen there is not only a sperm, there are some secretions of different glands, they're like prostate and bulbourethral, seminal vesicle, all these glands secrete some substances. For what? One thing is there is need of liquid, only then the sperm can survive. Another thing is there is need of nutritive substances. So that nutritive substance is what? Fructose. For other cells, nutritive substance is glucose. But for a sperm, it is fructose. Therefore, glucose, particularly in the testis, or exactly I am not getting in which cell it is converted, but particularly in male reproductive system, glucose is isomerized into fructose to provide nutrition to the semen, and it can be reduced in fructose can also be reduced in mannitol. I described mannitol. You have to ask something? Now, sorbage. Obtained from mountain ash, it is a plant, mountain ash. Sorbitol, obtained by reduction of sorbage. Therefore, again, reduction. Glucose is converted into alcohol by reduction. Slowly metabolized. No, it is not metabolized easily. There is slow metabolic activity of non-stimulant laxative. What happens? When we take laxative, what does it do? It increases the motility of the digestive tract. Have you heard? Recently it has happened. Ruchi went Delhi and she was suffering from diarrhea. She went to the doctor. Doctor suggested a medicine. She took the medicine. She was going to recover. Her father suggested, actually for these things, that sarbat of wood apple, way. Have you heard food apple? So if it is taken, then it cures. But wood apple, particularly sherbet of wood apple, helps when it is due to heat. If it is given in the infectious case, no, it is due to infection, it increases more. And within half an hour, she became nervous. When I called her, she was unable to speak. That time I became restless. I called many persons and deputed 2-3 friends, and then she was carried away, and she went, and then they carried her to the doctor, and then doctor gave another medicine, then it was cut. Because that wood apple contains many fibrous thing, cellulosic thing, and that acts as laxative, not only laxative, that is a stimulating laxative. It increases, particularly seed of wood apple, and some fibers present in it. The pulp contains some chemicals that increases the motility of the digestive tract, and therefore there is more. So used in sorbitol is used in bacterial culture media to distinguish pathogenic E. coli. Whatever we are using, DH5 alpha, it is non-pathogenic. Particularly when E. coli becomes pathogenic, then it causes a disease that is called colitis. In our intestine also it is present, but it does not. It is symbiotic. But sometimes what happens, there is certain mutation in those symbionts, and then it becomes pathogenic. So in a culture, if sorbitol is added, at that time it is distinguished, it is present there or not. That is it is also, ketone is there, therefore it is ketone. Now, psychoge, known as rare sugar. Energy generated from psychoge is very low, therefore ultra low energy. Can be obtained from the antibiotic psychofuranin. Therefore its name is psychoge. Okay? It is obtained, can be obtained from, actually first time it was extracted from the antibiotic. Psychofuranin. Helps in controlling hyperglycemia, hyperlipidemia and obesity. Hyperglycemia means high blood glucose level. Hyperlipidemia means high lipid level, and obesity. As you know, when there is high lipid level, then it will accumulate, and different parts will expand, isn't it? That is obesity. In this case also there is functional group is ketone, therefore it is ketose.